It seems like
right side downwards.
so we are looking to move for the next remaining topic in this unit for the preservation. As you are doing some cultures, right? We need to go for some preserving the bacterial suspension, fungal, or any microbial suspensions are there, right? So there are several basic techniques are into it for the maintenance and preservation. What is the first part? Why we go for the any preservative methods? Some basic reasons you can say that to do the same work continuously. Sometimes you are working, we need to do the same culture, like E. coli. For the basic experimentation is there for the general class. Or it is maybe you are working on some particular targeted work. Okay, then it may be a beneficial or harmful. It is like a pitching or you are making the buttermilk. For the buttermilk, we need some lactose. So every day, if you are working in the dairy microbial, dairy-based institutions are there, dairy microbiology, you are doing, then you will need the one culture always. Then it may be it is like you are working in the medical sector. Okay. Then you are looking at if there is antagonistic effect is there or not, or any parasitic effects or not. So to study them also we need some pure culture. Even then it is like a new disease has been spread. So we need to do this in experimental part. So in any case, it may be beneficial or harmful organism there. We need to go for their research. Maybe right now it's not required, but in the future we may go for it. So isolated organism needed for lab-based class work, research work, as a test agent for particular procedures. Sometimes we are just looking that E. coli is the sensitive or not, or some bacterial suspensions are there, colonies are there, are sensitive to certain antibiotics or not. So for such methods we may go as a reference strain for the taxonomic study. That means some organism is been observed, so to match the characteristics, we may go for it. Yes. Yes. So biological companies, there are the several institutions are there. Initially, it was a job of just a researcher. When a laboratory they are doing, and it's a master degree, what's going on, they will store it for a while. Then their work is done, they will give to their students. After it may launch. So initially it was done in an institution level. Then after so many companies and the you can say the culture collection centers supported by the government is also being started. If you are looking at in India also there in abroad everywhere it is the culture collection centers are there. You can submit over it. There the minimum of charges are there. So this institution now is going to do a subculture or maintain your given center. There are several methods are there by which we can do the preservations. That part we are going to learn in this process. So basic methods, you can go for like a basic simple method, and one is a sophisticated. Simple method is nothing like into it. You can go for like periodic transfer. Yesterday, we have done some substances right, how they are shaping those. Well, this is some basic single column. Now you may pick up that column, transfer to the plant, or else into nucleus. So if you are going to stick again and again, so you take a periodic transfer. That means same value there, but you can transfer to one to another. And you must store at a low temperature in a freeze. So it will be the metabolic activity is going to reduce down. Basic characteristics here is what? To restrict their growth. Otherwise, they are going to deplete your all the microbial nutrients given to it. If it is a feeding is allowed, they may grow. There is a no harm with the growth. But they are continuous to growing. One day, the microbial even medium is going to get exhausted. As exhausted, afterwards they may die because there is a starvation. That means no availability of food and then it is kept in a room temperature or 27 degrees centigrade. So we need to retard their process. So what we have to do? Go for it. We will do. We transfer them to refrigerator. This is some basic techniques. Other methods are there. We are going to learn like oil overlay. That is like a whenever the medium is there. Okay, you have done the. You can prepare the slant. After the slant is prepared, you have just inoculate sticking this slant. Then after you may transfer the mineral oil over it. As the mineral oil does not allow continuous growth. Okay, it will prevent also availability of oxygen. The growth will be somewhat retarded, but the bacteria is trying to manage it. Okay, so nutrient is available. So over a certain period of time, you need to just store into some basic refrigerator. So with also basic overlay, but there is certain disadvantage. Sometimes the stain may get mutated. The mutation is what? It is spontaneous. Okay, the spontaneous mutations may arise. The reason is what? That you have stored it. Its nutrients is available, but you are not providing the continuous aeration. The macros may start to become a faculty. A certain enzymatic process may initiate it. Cell divisions may be retarded, but it may cause a certain mutations. Then the soil culture. So one by one, certain methods are there. Then the sophisticated is a glycerol stock. Then liquid nitrogen and lyophilization. So one by one, we will go for it. Okay. So periodic transfer, which is well known. All the majorly at a basic level laboratories are doing it. In the periodic transfer, monthly, generally it is required. Sometimes or twice a month, or 2 months, 6 months, as our capacity. We have to check it out whether the plates are depleted with given microbial material is there or not. Nutrition availability is depleted. One thing. Another, you have to check it out continuously whether it has any contamination or not. If you are keeping in a freeze, sometimes power fluctuations may happen. It's usual. Okay. Due to the power fluctuations, the temperature may have risen up, and the transfer of the from here to there may happen, right? So one has to be take care about it. So one has to take care about it that whether the freezing during the freezing, the power loss is there or so. Some advantage are there within it. Fine. So the stored in properly. This happens that because the metabolized nutrients present in the medium, hence after some time nutrients level may decrease. And yes, another problem during the periodic transfer is what? It may have some toxic compound release. It is like their extra material. It is been released by itself by the microbes. Okay, it is required to get taken from it. Right. It is like you are in a prison also. If you have seen the certain movies are there. Okay. So in a prisons also, what is being just provided? Just maybe I have told earlier also, right? Some bed-like structure is there. Within that, it is the one tap is there for drinking and every sort of activity. Even there is a for the one, you can say the like a round like part is there for there going to toilet and latrine. Within that, one has to do every sort of things. Okay, that is the whole day. For a certain time period, they may take in out for their bath. It is a common bath. It is like a you may have the hostel facility somewhere. It has been seen. So here, the toxic part, if it is the nourishment is given continuously, feeding is given, right? But the excreta, it's been taken out. If it is not there, if it is lying there itself, the person in the prisons also may help get curious. So we are maintaining it. Okay, we are just torturing in one to another way. Reason is given, but it is limited and different. Okay, but here it is the bacteria. For our purpose is a preservation, but for them is a imprisonment. Okay, it is a imprisonment. They are in stored or kept in the periodic transfer event, but they are releasing some extra toxic compound. Excretory part is there. Second, metabolites or other metabolites have been released. It became toxic for it, and they may retard their own metabolic process. So we need to go for the continuous periodic transfer. How it is generally been done, like a nutrient agar slant is used. Why we are using the slant? Can anyone have any clue? Why we are generally not preferring any plates? Rather, we may go for the slant to be get used while in preservation. Okay, chances of contamination because of surface area. Okay, fine. Any? Yeah. Yes. Yes. So it's a concern with the areas and the surface areas or like that. When the plates you are opening, there is a more chances to exposed with the air. While we are opening the tubes, okay, it is between the two burner. Okay, and another thing is we may just their mouth part. What we can do, make it sterile, or we can just what it. Okay, we may pass through the fumes. Then it may be does not allow to another microbes while opening or the subculturing to be get moved. And another most important part is that the storage volume required lesser. To store the plates, it may require more spaces in a freeze event. If you are working with some 100 microorganisms, 100 isolates are there. For that, you will require the one all the segments or the one compartment of these to be used for the storage purpose. If you are doing on a plate or in a tube, you can make a bunch. Within the small segment area, you can store it. So one is the surface area and second is the storage volume. Can be reduced while using the slant. And third advantage, it became easy to handle. Right? Is this became easy to handle, economic, and other blah blah things are there. But major two things are. So stock culture, primary, secondary, we may incubate for a period and the slant stored at a 4 degree centigrade. In a freeze, we may store at a 4 degree centigrade. It's normal. And we may go transfer inoculate the microbes rest medium after a specific time. Maybe it's a 1 or 2 months. Over there also we have to check it out. Sometimes may happen. It also get contaminated. There are so many reasons behind it. Maybe a handling error or any power fluctuations is there. So stable power supply is required. It's a mandatory when we are working with the preservation. While working in this area, microbial work is going on. So during the present preservation span, stable power supply is the mandatory things, and maintenance of every instrument is also required. Sometimes we may go for the continuous monitoring. There are AMC is also taken. Okay, AMC. Do you know about what is AMC first? AMC. For any instrument, AMC is must to be taken. Annual maintenance contract. That some instrument you have taken, right? Maybe it is from the here or the foreign institutions, foreign companies are there, you have purchased. So annual maintenance contract is required. They periodically coming, maybe a 2 months, 6 months to check it out, whether it is working properly or not. Calibration is required. Otherwise, we may think that okay, temperature is going on, but the real temperature is not there. There may be some technical errors. Disadvantage, just maybe it is like a microbes have to subculture time to time, and microbes are frequently subcultured. Mutations can occur. That is the but obvious. Whenever you are doing the subculturing, chances of contaminations maybe is there or time consuming. Another problems we may say that frequently subculture leading to sometimes mutation. It is a spontaneous mutations. We may know, we don't know. Maybe it is a harmful or beneficial, but there is a chance of getting mutation. I think in whole, I have kept small document how it is actually done. Here it is related to culture transfer and the subculturing step. How we may go for it. If you are having your mobile phone, you can start documentaries to be get listened along with this. To prepare the test for the subculture for the bacterial preservation. This is some well known. Under aseptic conditions, what we may need to do, wear the gloves, wipe the area which is disinfected. You may prepare the area with in laminar airflow or in a between two burners. Subculturing aseptic transfer, label properly, which is just some basic techniques. Okay, we are currently not going to follow it or perform. It is like a transfer from one to another. I think this video is not having any voice. I'm not sure.
Sir, no.
You can mute it. So here you can see that how looping is done, right? One was having the bacterial suspension, another was not. It is like a subculturing from one to another. Continuous subculturing is required. How we can do it? Either you may go in a flat even or in a tube. The same part you may have done earlier, that is like a subculture, we have given the broth, right? If it is given the slant, you may do again in the transfer process. Just this. Okay, just next part is given, that is how it is been performed. So here is this person is regularized and showing it how to do. So maybe he is using some flex between two burners. He may start. It's like sometimes scraping just. Colonies are there. Using the envelope, you scrape it and transfer to another. So how many times he is making, he or she is making the line, you can see. sector wise they are doing. Just so many time it is a they are making streaking. It's coming with a practice every time. He's crossing the line even. Then after it is reduced. Okay. It's going to happen. Yeah, no problem. It is like just subculturing from one to another place. So what they are doing, they are using the another loop. So one has to no need to wait for a continuous, that loop get sterilized and pulled down. So how many times striking is then? Fine. Another is a like, huh? But સુકાઈ નો વાત નથી. ફટાફટ એટલે we no need to wait for a continuous hours. It should be done within a given time period. એટલા માટે જેને શું કર્યું કે 1 થી 2 વાયર લૂપ યુઝ કર્યા. થોડીક વાર પછી કરશો તો બેક્ટેરિયા તો ત્યાં છે જ, તો પાછું વાયર લૂપમાં ચોટવાનું જ છે. એમાં તો કઈ ફેર નહીં પડે. હા એટલે જ આપણે ગરમ હશે વાયર લૂપ. પેલી વસ્તુ વાયર લૂપ હોય ગરમા ગરમ દજાડી દે તો શું અગારને તમે ગરમા ગરમ અડાડીને ટ્રાય કરજો, કઈ નહીં થાય એવું તરત. કારણ કે અગારને મેલ્ટ થતા જ વાર લાગે. ઇટ્સ રિક્વાયર્ડ અ લોટ ટેમ્પરેચર. એટલું નથી એમ. ગરમ ચા હોય હાથમાં અડે તો આપણે બળતરા નથી થતી એટલી, બરોબર છે. કદાચ આંગળી ઉભા છે એ જ વસ્તુ જીભને કેમ તકલીફ પડે? ઓર્ગન ઇઝ સેન્સિટિવ, બરોબર છે. એ જ વસ્તુ તમે પામ ઉપર અને અહીંયા ચા ઢોળાય તો વધારે ગરમ લાગશે, અહીંયા નહીં લાગે. ધીસ પાર્ટ ઇઝ નોટ ધેટ મચ સેન્સિટિવ. It can tolerate. So bacteria cannot. Simple reason. Oil layer, yeah. Here the 1 inch layer is done. About 1 inch of mineral oils, sterile mineral oil we may use just to overlay it, and can be stored up to 6 months. Here the prevents dehydration of media. One of the advantage is what? That prevent dehydrations, because if it is kept in the longer time period, even in refrigerator, we know that the refrigerators are coming with a defrost. What is that defrost? Anybody having any idea? What is the defrost? ઘરે કોઈને ફ્રીજ છે? Okay. Single door, double door? Double door. What happens single માં? જામી જાય. ડબલ ડોર માં પાણી. હા, ડબલ ડોર માં ક્યારેય જામતું જ નથી, સિંગલ ડોર માં જામી જાય છે. રીઝન શું છે? નાનું નાઇટ્રોજન ક્યાં આવી ગયો? એ હોય ક્યાં? Do you know the difference in single door and double door? Maybe it's a multiple door, but more than one door freeze is there. Yes, another. If you are going into a certain place for the purchase the ice cream, it having the whole just freezer. This may be a vertical or a horizontal. Horizontal, vertical freeze is there. If you are open up, whether it having ice within it, if you are going in the place in the market to purchase the ice cream, you may open up, right? Open the doors. Maybe it is a horizontal. They are taking out the one box of ice cream, right? It may have the ice within their border side. ઘણી વાર હોય છે. Seen? You may go to the Amul markets anytime here, backside. Anytime purchase any ice cream or Amul parlor. Have you seen it? क्या शौक है ice cream खाओ? So the thing is what, that their doors are, there are the two modes are there. For defrost, it's continuously on the or you can say the surfaces are the backside. If it is continuous temperature is increasing, the water is going to get frozen over there within their party. So what need to do? It is going to get solidified and the crystallized form is going to happen over it. Continuous in the double door phase over the on the wall, it may have the within it a metal sheet part is there within it. Don't put anything, you know. I'll kick you out again. After a few days, just some instructions maybe get forgotten. Right? So it is like a low temperature. Okay, it is maybe at after certain time period it may start. So their temperature maybe it is at 15 to 20 or 25 they may start. So whatever ice been maybe started to what get crystallized or ice formations maybe started, it's get melt down. And within the melting, it may be the gas is taken up. Water vapor is taken up and it is get collected, pull it down at the bottom. So you may find that on the bottom, there is a one tray is kept. It continuously gets filled. So from the air, okay, from the air, if it is any water vapors were there, it is been trapped. If it is trapped, get crystallized, it also get melted down again, it is been taken up. So it is the basic phenomena it's going on. Sometimes in the single door, one switch is given to defrost. On that time only, it start to get their surfaces within it, oil is given. It start to get a water and it going to melt down the ice. Sometimes many people what they are doing, they may switch the defrost in the single door. Otherwise, they leave it open for 1 or 2 hours. It's going to get melted. Both the way it's possible. Previously, the buttons were not there. Nowadays, it's coming for the making a defrost. So these are the two different advantage and disadvantage of each thing. If you are using for the preservation purpose a defrost mode, any of a refrigerator is there, right? So what is going to happen? It continuously after it gets you can say their circuit has become the hotter, the water vapors were there is been taken up, sucked up. So continuously water depletion is going to happen. Whether it is in medium, there is a cotton plug is there, it is like a suction pump. Continuously suction pump is taking the place. And because of it, what's going to happen? That water vapors may reduce. As the water vapor may reduce, your medium, it is like a water or start to get transpiration. From the water medium, transpirations may happen via cotton plug, it's going in a freeze. Within the freeze, its sucking pump is going on. Suction pump is there. So after a certain time period, you may think that what the medium one freezer on the freeze, what we have kept, it became a like simple one thin layer. Papad જેવું થઈ ગયું એમ કહી. So why it's happening? That water has started to get depleted. If you are using this overlayer, oil overlayer is there, it prevents from the oil, water will not move out. So it can be stored for a longer time period, maybe about 6 months. Okay, same slant is there, overlayering is done. But the disadvantage is time to time need to subculturing even here also. Okay, and the microbes are frequently subcultured, mutations may happen. It is similar to the previous method, subculturing, but it is oil overlay, maybe for a longer time period we may store. So here one small documentary is there, let's see. The voice is coming.
2 या 3 हफ्ते के अंदर डिहाइड्रेट हो जाते हैं और हमारा जो
I
कल्चर
think
है
it
वो
is in
डेड हो
Hindi.
जाता है। पर अगर हमको
What's it?
स्लांट्स को थोड़े ज़्यादा दिनों के लिए, जैसे कुछ महीने, माइक्रोबियल कल्चर की ग्रोथ चाहिए, इसके लिए आपको स्लांट्स को स्ट्रीक करना होगा अपने माइक्रोबियल कल्चर से। उसके बाद आप स्लांट्स को करेंगे इंक्यूबेट। इंक्यूबेशन के बाद आप देखेंगे कि आपके स्लांट में ग्रोथ आ चुकी है, जो भी आपका टारगेट माइक्रो ऑर्गेनिज्म था वो उसके अंदर ग्रो हो चुका है। तो अब आपका स्लांट रेडी है ताकि आप उसको पैराफिन मेथड से प्रिजर्व कर सकें। तो ये प्रिजर्वेशन स्टेप बड़ा आसान है। आपको करना यह है कि आपको जो अपना स्लांट है उसके अंदर लिक्विड पैराफिन को फिल करना है। फिल कहाँ तक करना है? जहाँ तक कि आपका स्लांट की हाइट है, आपको
Yes,
उस
you can
हाइट
see
तक
that he is
लिक्विड पैराफिन को फिल
overlaying.
करना है। उसके बाद आप इसे कॉटन प्लग लगा करके इसको रेफ्रिजरेटर के अंदर आप प्रिजर्वेशन के लिए रख सकते हैं एक लंबे टाइम के लिए। यहाँ पर आप एक ख़ास बात देखेंगे कि जो भी एयर बबल्स स्लांट के अंदर प्रेजेंट है वो धीरे धीरे करके ऊपर की तरफ आ जाएंगे और लैप्स हो जाएंगे एटमॉस्फेयर के अंदर। इससे क्या होगा कि एक ऐसी कंडीशन बन जाएगी, ऐसी सिचुएशन बन जाएगी स्लांट के अंदर कि माइक्रो ऑर्गेनिज्म डोरमेंट स्टेट में चल जाएंगे और एक लंबे टाइम तक वो स्लांट के अंदर प्रिजर्व फॉर्म में रहेंगे। सॉरी टू डिस्टर्ब, लेकिन एक इम्पॉर्टेंट इन्फॉर्मेशन देनी थी कि
Okay. So what he is telling that continuous when you are doing the subculturing, we need to just incubate maybe about 6 to 8 hours or 24 hours. Just incubate, and afterward you may overlay with oil, mineral oils. Okay, there are so many oils are available in the laboratory that you can just overlay above it. So it's what going to happen that bacteria get not able to get the continuous. One important part is if it is anaerobic or the aerobic. That is also most important to understand. Aerobic bacteria will not get the oxygen conditions. They need to go in a very low and static condition, and it is freeze in a temperature with low. So they are maybe viable. Microbial nutrition is there. That viability is not lost. Viability means they are not died. If the again the process is done, it can be reculturing is required. So overlaying is basic method. Fine. Till this, anybody having any questions? Just culturing, subculturing, and the oil layering. Simple method. Yeah. So incubate it directly. If you are overlaying, then the microbes were not able to grow. So before overlaying, you may incubate for about 24 hours or 48 hours. It is up to you what the microbes are there. So we need to incubate. So now microbes is fully grown on our slant. Then you need to just you can say the overlay. Stage 1, stage 2, step 3 are refrigerator. Yeah, we need to cover. It is mandatory. Cotton plug. Another is also people are using the above the cotton plug even. Above the cotton plug, they may use the paraffin, one kind of like a elastic material is coming. So you may wrap it that part. Okay, so it also prevents the direct transfer of any sort of the microbes from here to there. Even on a plates, whenever you are using the plates, right? Plates are being wrapped with sometimes plastic. Taps are also different. Paraffins are also coming. So there are so many other people are using. Earlier people are using the just wax. Okay, the candle is there. Just you may put the drop down. It's like a ceiling with the wax. It's easier. The previous method. Okay, so it is nothing special into it. Another is a simple. It is just a wrapping. હા કરી શકાય. ન્યૂઝપેપર કર્યું હશે તો થોડુંક એન્વાયરમેન્ટનું કન્ટામિનેશન થવાના ચાન્સ ઘટે. ઘણા લોકો ન્યૂઝપેપરથી રેપ કરે છે. દરેક મેથડ ન્યૂઝપેપર રેપ કર્યું હશે તો 100% કન્ટામિનેશન નહીં આવે એવા ચાન્સ નથી. ઘટશે ખરા. એ તો કોઈ પણ વસ્તુ છે કે if you are going in outside, you can have the raincoat. Okay, rain suit is there. Different kind of rain suit you may use. You may use the umbrella. It is up to you. You want to how you are going to get protected in a rain. Another method is soil culture. Okay, soil culture, we know that the soil is basically having the some micronutrients already within it. ત્યારે શું ચાલુ છે અહીંયા ચાલુ કર્યું